You to persist, and it's always difficult to give the last lecture, so I'll try to go through it very quickly. I want to talk to you about breast milk and microbiota in the premature gut as a potential mechanism for preventing necrotizing enterocolitis. First, before we consider this, we have to consider the clinical problem, and then ways that have been used previously to prevent NEC, such as the expression of mother's breast milk and probiotics, and then a different approach, which is probiotics in breast milk-fed infants, before we summarize and conclude. Let's begin with the clinical problem. We all know how devastating necrotizing enterocolitis is. Even if the infant survives, it has lifelong problems. So it's very important that we prevent rather than treat the condition. 10% of babies under 1,500 grams develop necrotizing enterocolitis. It represents 1 to 5% of NICU admissions. In greater than 90% of cases, it occurs in premature infants, and it occurs very commonly 7 to 14 days after delivery at a time when the infant has been fed. And susceptibility is inversely related to gestational age. Now, this looks very familiar because it was taken from the same article, and Joe and I wrote a review article on that. This is a patient with necrotizing enterocolitis and pneumatosis intestinalis. 30% of patients with NEC require surgery. Surgery invariably leads to short bowel syndrome, and about up to 50% of babies, whether they're operated on or not, die of this condition. Sequelae are enormous and are lifelong. Short stature is very common. There's delayed neurologic development, particularly in cognitive function. All of these have a lifelong persistent problem, and the medical costs are prohibitive, costing about $2 billion per year. Several years ago, we hypothesized that necrotizing enterocolitis was caused by an immature, inappropriate, innate immune response to colonizing bacteria in the gastrointestinal tract. To support this hypothesis, we used organ culture of intestinal biopsies from newborns and then tissue obtained from therapeutically aborted fetuses, and we challenged these tissues with lipopolysaccharide and IL-1 beta, which are common causes of necrotizing enterocolitis. As you can see, the IL-8 protein level and the gene level was much higher in fetuses than it was in the neonate, suggesting that the excessive immaturity of the intestine was a factor in the inflammation. Now, the innate immune response, inflammatory response, occurs when bacteria interact with toll receptors, which trigger signal transduction molecules, such as MYD88, TRAFT, and TRAC, and it leads to a transcription factor which enters the nucleus and causes transcription of inflammatory cytokines such as IL-8, TNF-alpha, and gamma interferon. Now, under normal conditions, this is a self-limited process because there are other components that interfere with the signal transduction and inflammation that address different aspects of the signaling process. We decided we would look at the components of the innate immune response in mature intestine, in intestine from aborted fetuses, and intestine from the resected portion of necrotizing enterocolitis. And as expected, we saw a strikingly elevated IL-8 response in immature intestine and in the necrotizing enterocolitis patient. When we looked at the signaling molecules and the receptors, we found that in each instance, TLR2, TLR4, MYD88, TRAFT, and NF-kappa B were elevated. What was most interesting was that when we looked at the negative regulators of the innate immune response causing a control of this response, we found that mature intestine had higher levels and immature intestine and necrotizing enterocolitis considerably lower levels, which we felt contributed to the hyperinflammation that occurs in necrotizing enterocolitis. All right, with that brief introduction, let's consider approaches to the prevention of this disease. We know, and Joe has already talked a little bit about that both breast milk and probiotics have been used to treat the condition. This is that meta-analysis that Joe referred to, showing that in small numbers of cases using different probiotics at different doses, that there appears to be an improvement. But you heard the other aspects of that that he discussed, so I won't raise that right now. There's also a meta-analysis of breast milk studies. This is mothers express breast milk fed to the infant. And as you can see, in each instance, this was very successful. Now, we know and appreciate that breast milk influences the development of mucosal immunity. It does so through the microbiome, which we heard about earlier today. It does so through protective nutrients such as prebiotics and anti-inflammatory factors such as lactoferrin and omega-3 fatty acids, which we'll hear about tomorrow, and immune-enhancing substances such as nucleotide. What you might not appreciate is that breast milk acts as substrate for colonizing bacteria to produce metabolites that are anti-inflammatory. Now, we did a study looking at the influence of breast milk, infant formula, and then the combination of banked breast milk plus infant formula. As you can see, this is different in each of these circumstances. We also did whole genome sequencing, and we recognized that the genes from breast milk in the microbiota were different from those of formula. So we decided to look at what happens when probiotics or colonizing bacteria interact with breast milk. The reason we did this was we knew that tryptophan-rich foods that are fed to animals or adults can produce metabolites which are anti-inflammatory in the intestine of experimental colitis patients as well as the actual IBD patient. We also know that when high fiber and complex carbohydrates are fed to individuals or into animals, that these, under the conditions of bacterial colonization and certain bacteria such as Bacteroides fragilis can produce short-chain fatty acids, which by a variety of mechanisms are important in gut homeostasis. For example, fiber fed to animals produced short-chain fatty acids. These short-chain fatty acids interacted with G-coupled protein receptor, in this case 43, that are present on T regulatory cells, causing an increase in the expression of these cells. This is the microbiota itself, and these are specific and mixture of short-chain fatty acids, all of which cause an increase in T regulatory cells, as opposed to germ-free that has no increase at all. It also caused an increase in IL-10, which is an inflammatory cytokine. Now, we were most enthused about this approach to prevention when we noted in a paper that appeared in Pediatric Research that when probiotics were used with breastfed infants, they were much more effective than used with formula-fed, and depending on the degree of breastfeeding, it increased the dependency on preventing necrotizing enterocolitis. We had previously shown that Bifidobacteria infidus secretions free from the bacteria were anti-inflammatory. And what we did was we separated bacteria from the media itself. We then used fetal intestinal cells, which we pre-incubated with various fractions of the media, and we showed an anti-inflammatory effect that occurred. Now, when we used the less than 3K and 3K fractions in collaboration with the group in Salerno at the Research Institute in Salerno, we found that the 3K and 3 to 10K had a peak that didn't exist in the media. Now, using a database, we found that this was indolactic acid, which is a metabolite of tryptophan. We know that breast milk has large quantities of tryptophan, and so we felt that this was a potential source of anti-inflammation in the patients with necrotizing enterocolitis. What we did was we repeated the studies using fetal cell line, incubated with various concentrations of indolactic acid before we challenged with IL-1 beta. And as you can see, there is a striking reduction in the inflammation. To confirm our in vitro observation in human fetal cells, we used intact mice, both fetal and mature, and found that we could repeat this approach using fetal mice, but it was not the same as mature mice, suggesting that it was a specific fetal effect. Now, we know that indoles from tryptophan interact with aryl hydrogen receptors, which can function as a transcription factor to produce IL-8. But when we used an inhibitor of these hydrocarbon receptor, we found that we lost the anti-inflammatory effect, strongly suggesting that this was the mechanism by which this occurred. Now, let me try to summarize what we have shown here. We have shown that when breast milk, which contains a large amount of tryptophan, interacts with Bifidobacteria infidus, this produces indolactic acid, which is then transported across the fetal enterocyte, interacting with the aryl hydrogen receptor and causing a downregulation in inflammation. We also know that complex carbohydrates in breast milk interacting with certain bacteria such as Bacteroides fragilis can produce short-chain fatty acids. What we didn't know was, do short-chain fatty acids have an anti-inflammatory effect on fetal enterocytes? So what we did is we incubated different concentrations of acetate, propionate, and butyrate, and found that each caused a reduction in inflammation, suggesting, again, an anti-inflammatory effect. When we looked at the mouse model, we found that the anti-inflammatory effect persisted in the small intestine as well as in the colon. And looking at the mechanism, we know that short-chain fatty acids affect deacetylases, causing a reduction and therefore reducing their epigenetic effect on the inflammatory cytokines. So here we show that when we used the 3 short-chain fatty acids with 2 fetal deacetylases, that in both instances they reduced the effect. We also know that short-chain fatty acids work through G protein coupled receptors. And in the case of the neonate, this is a 109A G protein receptor. And what we showed is that when these short-chain fatty acids were incubated with fetal cells, that they caused an increase in this receptor, and that when we used an inhibitor of the receptor, it suggested that we lost the inflammatory effect, suggesting that 109A was the receptor for this process. Now again, let me try to review with you what we've shown. We've shown that IL-1 beta, which is a common stimulus in necrotizing enterocolitis, can pass through its receptor and cause an increase in deacetylases, which by an epigenetic effect causes an increase in IL-8 and increased inflammation. Short-chain fatty acids through the 109A receptor can cause a reduction in the deacetylases, less of an epigenetic effect, and therefore affect inflammation. So these are 2 examples of how bacteria acting with breast milk in a different capacity can produce anti-inflammatory effects. So let me try to summarize and conclude what I've said. I've said that necrotizing enterocolitis is a devastating disease that must be prevented and not treated, that the best way to prevent this is to use breast milk in conjunction with a probiotic, and that although breast milk prevents NEC by a variety of mechanisms, including oligosaccharides and positive substrates, it also works by producing anti-inflammatory probiotics, metabolites. And then I've suggested that breast milk tryptophan metabolizes B. infidus, metabolized by D. infidus, produces indole-3 lactic acid, which is anti-inflammatory, but only in the fetal intestine. And also that breast milk interacting with other substances can produce metabolites like short-chain fatty acids, which are anti-inflammatory. Thank you very much. Questions? Yes, Jerry.
Allan, you listed lots of different compounds in the milk with anti-inflammatory activities and so on. How about IgA? Do you remember Martha Abel in Vienna?
Yep. No, but I'm now interested at this point in how probiotics metabolize breast milk. IgA may be a very important component, but I'm just looking at that side. Any other questions? Ray?
What is the
Okay. Yeah, okay.
Do you see necrotizing enterocolitis in breastfed babies at all?
Yes.
And at what level do you see them?
Yes. Now, let me preface by saying I'm not a neonatologist.
Me neither.
Joe New is, but I'm trying to work out ways that we can do some preclinical studies
Right.
that can then be done in large clinical studies that hopefully will help prevent NEC.
And the second question I had was, are there any animal models in primates where under natural circumstances you see NEC?
Joe? I don't know of any.
No, but I think the closest one is if you take, for example, a piglet, and if you do not give that piglet IgG, and you artificially feed that piglet, where you do not allow any mother's milk to that piglet, within 1 week that piglet will develop a bloated intestine, and it looks like necrotizing enterocolitis.
You know,
It's
Joe,
not been called.
piglets are really different from humans.
Right.
Yeah.
They
In
don't
fact-
have any immune
Particularly
function,
pigs,
and they
yeah.
require breastfeeding to get function. So I don't think that's a
Well,
reasonable
but
model.
I think that's the question you asked.
Yeah.
Yeah.
And the third question I had again is related to the underlying pathophysiology of what we call NEC. You know,
Yeah.
what is the role of hypoxia in this situation? Because we are looking at a tube, which creates a lot of holes, and you put a lot of air out of it. I thought there were some early observations which suggested hypoxia may have a very important role in the induction of.
Yeah, I agree with Joe that this is a very complex disease and that there are lots of factors that are involved. Hypoxia has been known. I'm just looking at one aspect of it.
But couldn't you explain a lot of these things which you see purely because of cellular damage? All the IL-8 and other lymphokine secretions may be purely related to the cellular damage rather than
Well, as I
changes in
showed,
microbacteria.
you have
and-
excessive IL-8 in necrotizing enterocolitis and immature intestine. So I think those are factors that are involved.
Right.
But I don't mean to imply that the only situation is an immature innate immune response. But it is true that in human prematures and in necrotizing enterocolitis tissue, that this is the case in terms of an immature innate immune response.
Are there some characteristic features of microbiota composition and function, it means metabolites, described in infants with necrotizing colitis?
There is. A number of people, including Joe New, have shown that there is a change in the microbiota before necrotizing enterocolitis occurs. And he explained that in his talk, that there are changes in the microbiota prior to the institution of necrotizing enterocolitis. Isn't that true, Joe?
It is not connected with some real characteristics. It means that there is existence of some species which are not in healthy premature children.
Well, is it okay?
Go ahead.
Yeah. That does seem to be the case, that you have a dysbiosis with a shift in the two different phyla One is an increase in proteobacteria, which is largely the gram-negatives, and the other one is a decrease in the firmicutes, which is gram-positives, those bacteria that are butyric acid producing, some of the same metabolites that Allan was talking about.
Now, we have previously reported that the commensal bacteria or bacteria that are good bacteria can cause inflammation in the fetal intestine. So I think that's part of the problem. The response is abnormal.
One thing, in case of preterm low birth weight, whether any early feeding can give rise to the necrotizing enterocolitis, any feeding problem that related to the necrotizing enterocolitis?
Yeah, I'm not a clinician. So what I'm doing are preclinical studies. And before we can recommend what we do with preemies, we have to do a clinical trial. And that's something that has to be done. I'm just pointing out preliminary observations that suggest that breast milk in combination with the probiotic may be effective in creating an anti-inflammatory effect.
Okay.
Yes?
Yeah. Do we consider the difference between the real breastfeeding and breast milk composition? Because I think, okay, maybe the real breastfeeding is also different from the breast milk because there is some skin contact, maybe benefits for the premature immune system.
Go ahead, Joe. Do you want to talk?
So, I did bring that up a little bit in the talk that I gave, where the skin-to-skin contact and the enteromammary system that was described so nicely in the late 1970s by Allan and colleagues. I think that does play a role, and it's. But as far as the evidence is concerned behind that, I think that we still don't have hard evidence. We have good associations. We have the studies that were done in Israel, also in Western Australia, by Hasatu and colleagues, where it appears that this enteromammary system actually does occur, which necessitates close contact between the mother and the baby. And this is one of the reasons why in neonatal intensive care units in many parts of the world, we're trying to encourage the mothers to come in and to snuggle with the babies, have the babies on their breasts, and not just feed them the milk itself, but to have that direct skin-to-skin contact.
I should point out that mothers' expressed breast milk is far superior to banked milk because it has so many different factors that aren't gotten rid of when you pasteurize. And actually, mothers' breast milk from breast banks are later breast milk, and it doesn't contain a lot of the very important protective factors that exist with colostrum and mothers who deliver prematurely. Any other questions? Okay, we have ended early because we are short one of our speakers. And Natalie, do you have some comments to make?